So yes, I'm very pleased to be here. I don't often get up here to the university, so it's fun to visit my department. I spend almost all my time at the HUD, so it is nice to be here every now and then. So this is the title of my talk, but on the next slide, I actually decided that this is really the focus of what I'm going to speak on today. Normally when I'm going to scientific conferences, I'm really focusing more on the nitty-gritty of the details of what I work on. But I think for this audience, I thought it would be better to make the presentation a little more broad. And oftentimes, whenever I tell people I work for the HIV Vaccine Trials Network, their first question to me, well, how's that going? What is the likelihood of getting an HIV vaccine? So this talk sort of brings together what is the current status of HIV vaccine, mainly from the perspective of clinical testing, from those vaccines that have reached the point of being tested in humans. And I think by the end of this talk, I think you should have a current understanding of where we are in terms of what's in the clinic right now, or about what is about to become into the clinic, because we have a big trial that's about to start. I should say we actually first enrolled our first participant just a few weeks ago in South Africa, and I'll finish up discussing that. So what I'm going to go through today in my talk is I'm going to first of all just introduce you to the HIV Vaccine Trials Network. That's where I work. Then I wanted to start with an overview of HIV vaccine efficacy trials that have been conducted so far. There are only a handful of them, so I think it's useful just for you to be aware of where we stand with what's been done so far. And then there has been only one of those trials that has shown any efficacy. That's the RV144 trial conducted in Thailand. That's been many years now since we've gotten results of that in 2009, but we're still really focusing our field on the results from that trial. So I'm going to spend a bit of time talking about the correlates that we learned from that trial. And then at the very end, I'm going to be talking about where we're moving on in South Africa, testing vaccines that were modeled off of that partially efficacious vaccine. And in the process of talking about RV144, I'll talk about cellular immune correlates, because that's what my laboratory focuses on, the cellular side of things, rather than the antibody side. And then polyfunctionality of the cellular response, which has proven to be a nice correlate in RV144 and is continuing to be useful as we go forward. And then I'm going to finish up by just reviewing the types of HIV vaccine candidates that are currently in testing. I think that's useful, again, just for you to be updated in terms of what's in the clinic and what's coming down the line, and which of those perhaps may at some point prove to be efficacious. And then the next steps in HIV vaccine efficacy testing, we're just very excited that we just started a new efficacy trial in South Africa, and I'll just briefly talk about it at the end. So I don't think I really need to focus on the broadness of the HIV epidemic, but I think all of you are aware of how large this problem is worldwide. And despite the fact of effective treatment, we're just not reaching treatment to all the people that need it. So I think it's clearly evident that we need a vaccine. A vaccine is really the best modality to control this epidemic, and I think the numbers speak for themselves. You know, obviously the biggest problem is in sub-Saharan Africa, and that's where we're focusing many of our research efforts. But we continue to have problems in selected subpopulations here in the United States, and it continues to be a major concern. And as I mentioned, even though we have very effective antiretroviral therapy, we're just not getting it out in high enough proportions to the people that need it. And that's really a major issue. And it is sad that we can't do that, but it's getting better over time, and I'm pleased to see that. But we really do, a vaccine is really what we need for preventing HIV. So the network I work with, the HIV Vaccine Trials Network, has this mission statement here. It's really to characterize the safety and the immunogenicity and the efficacy of HIV vaccine candidates. We don't develop the vaccines ourselves. We're a large testing network. People come to us to test their vaccine candidates. But we have the infrastructure necessary to do that testing. So we get vaccines from many different people. We have them from corporations, we have them from private entities, we have them from industry, we have them from government. So they come to us and we test them. So that is really the focus and the purpose of our network. I work within the laboratory laboratory side of the network. So we're focused on developing the assays to measure the immunogenicity of those vaccines. And hopefully, as we get efficacy in some of our trials, then to look for the correlates of efficacy in those trials. We have a number of international clinical sites. I mentioned many of them right now are focused in sub-Saharan Africa because, of course, that's where the infection is largest right now. We have a site in Europe, we have a site now in China, and we have 2 sites in Peru. And then among our domestic sites, we have a number of sites in the US as well. So it's a fairly large network. And in terms of the laboratories, we work on a centralized platform where we find it's most efficient to have central laboratories so that we have many different clinical sites, but then samples are drawn at those sites, stored in central repositories, and then shipped through our central laboratories. Our main cellular laboratory has been here in Seattle, but a few years ago we established a laboratory in Cape Town. We thought it was critical, since we're doing so much work in Southern Africa, that we have a site in Southern Africa where we can do our assays. And we're very proud of that lab. We have about 12 people in the lab. The director is American, but everyone else in the lab is African. So we're very proud of the fact that we have this lab up and running, and it's working quite well. I usually visit the lab once a year and spend some time there, but we're very happy to have a laboratory in Africa. In terms of just briefly what our laboratory research goals are, again, we're the laboratory component of the HVTN, and here in Seattle we focus on the cellular aspect of the immune response. Our laboratory in Duke and our southern and another laboratory in Africa in Johannesburg focuses on the antibody response. But for us, we're very interested in fully characterizing the cellular immune response to our candidate HIV vaccines, both so that we can compare immunogenicity across vaccine candidates. That's very critical. When you're testing a large number of vaccines, you need to determine, well, which one is better than another. At some point, you need to make decisions, and at some point, the government needs to make decisions in terms of which vaccine candidates are they going to continue to fund and continue to bring into the clinic for testing. And one of the ways of determining that is to look at immunogenicity. So we're very proud of the fact that we have standardized testing to allow for comparison across different vaccine trials. And then, of course, what we're most hopeful for is if we're lucky enough to get efficacious vaccines, then we can use samples from those vaccine recipients to look for correlates of vaccine efficacy. And that is ultimately the goal. Most of our vaccines have not reached the stage of efficacy testing, but we're hoping that more and more of them will over time. Our lab focuses on the development of very sophisticated immune assays. Many of them are based on flow cytometry. That's where my expertise is. And we perform these assays in a good clinical laboratory practice, GCLP. So not quite GLP, but very close to GLP. With the types of functional assays we're performing, GLP may be just a little bit too high a level to aim for in terms of what we're doing. But I think we're very close to that. So then moving on to, well, what do we need to get in terms of an immune response when we're thinking about an effective HIV vaccine? So most of the licensed vaccines that are out there for other organisms are inducing neutralizing antibodies. These are antibodies that neutralize the virus or the pathogen and prevent it from causing disease. So that has been for the long time an ultimate goal of HIV vaccination. But with the RV144 trial, and I'll just briefly refer to that in a few slides, we discovered that one of the correlates of efficacy there was a non-neutralizing antibody. So it may be potentially possible to have antibodies that are not neutralizing, but yet that are still effective in preventing disease. So these are antibodies that are going to be working through other mechanisms such as ADCC, so maybe perhaps through the FC portion of the antibody rather than the variable region. So that's very encouraging. So now we're thinking and we're looking both at neutralizing functions and non-neutralizing functions in terms of antibodies. And then, of course, we also feel that it's important to have a protective cellular immune response. CD8 cells, of course, for preventing, for killing infected cells, but CD4 cells will be very important for helping the antibody response. So whenever you have an antibody response, you assume you need a very helpful CD4 response as well. So we feel as though an effective vaccine would include all of these components, and we hope for that. So the efficacy trials that have been performed to date, there are actually only 6 of them, so there aren't too many of them. They've been performed in different geographic locations and also different risk categories. So it's important to keep that in mind. We may actually need to have different types of vaccines depending on the risk category. You know, if you have sexual transmission versus intravenous drug use, you may have to have a different type of vaccine. We don't know that yet. So when we're testing vaccines, we have to be careful to test them in a certain category and look for correlates based on the type of infection risk. And then these 6 vaccine efficacy trials have used 4 different vaccine immunogens. And throughout my talk, I'll be talking a bit more about these types of vaccinations, and I'll end on that a bit as well. So if we just skip ahead to just this slide here, I just want to really focus on one of these trials mainly. The RV144 trial conducted in Thailand was actually a very large trial. It was over 16,000 individuals, and these were not high-risk individuals. These were individuals just at normal risk of acquiring HIV. When the trial was first designed many years ago, unfortunately, there was a high incidence of HIV in Thailand. Luckily, while the trial was conducted, that was severely decreased, so the infection risk was relatively low. But yet there was enough information from this trial, enough HIV infections, to see that there was a somewhat protective effect of the vaccine. It was 31% efficacious. So that's modest at most, but still very encouraging. And when I tell you about the correlates analysis that have been done, I think you'll feel convinced that even though this efficacy was modest, because we're able to correlate that with certain immune parameters, we feel very confident that, that it was truly efficacious in a small subset of individuals. The very last trial listed here, HVTN505, was a study conducted here in the USA. We were very discouraged that we did not see efficacy with that. That was a prime boost types of vaccine candidate where we prime with DNA and then come in with a recombinant adenovirus encoding for HIV proteins. We did not see any efficacy there. We do have actually one slight correlate signal, and I forgot to include a slide on that, but a colleague of mine, Holly James, is submitting that for publication. So even in non-efficacious trials, we can still look for signals of correlates because in subgroups of individuals. I will, again, refer to that back when we talk about the RV144, because the correlate that we're finding in HVTN505 was similar to the correlate that we found in RV144 in terms of a polyfunctional T-cell response correlating with lower risk of HIV infection. In RV144, it was a CD4 T-cell response. In HVTN505, we saw it in CD8s. So it's encouraging that there is somewhat of a signal we're seeing. If we can build upon that, maybe we can move towards a fully efficacious vaccine. So what can be learned from human HIV vaccine efficacy trials? First thing I want to say right off the bat is that, you know, we do learn quite a bit from non-human primate testing. You know, that's really the only effective animal model with HIV. But unfortunately, it has not been predictive all the time. So I think it's absolutely critical that we bring these vaccine candidates into human testing. We've just learned that over and over again. So we try our best. We get some initial signals from non-human primate testing, but ultimately we don't know for sure that we're going to get the same signal in humans. So it is critical to bring them into human testing. But what we can do is from these efficacy trials, we can identify hypotheses to test for immune mechanisms that can mediate protection against HIV. So oftentimes, and what happened with RV144, many of the analyses of correlates that happened after the trial was finished are now post hoc because we didn't predict them ahead of time. We didn't really know what to look for ahead of time in terms of correlates. But now that we've determined some of these correlates, now we can do some further testing. We can design trials to see whether or not that correlate is indeed a correlate of immune protection. So we're really identifying hypotheses in many of these efficacy trials. And then identify biomarkers, and that's very critical for success of advancing vaccine candidates. The reason that's important, as I mentioned before, is you may have a dozen HIV vaccines that are in clinical testing. How do you rank them? How do you compare one versus another? So if you can identify biomarkers to look for, then you can see whether those biomarkers are actually induced by the vaccine candidate you're testing. So here's just another slide, once again summarizing different types of immune responses. On the left side, thinking about the antibody response, most often we're thinking about an antibody response as preventing HIV infection. We refer to that as sterilizing immunity, that if you give someone a vaccine, they won't become infected at all with HIV. So that's an ultimate goal, but that may not be feasible with HIV. We don't know that for sure. We may get non-sterilizing immunity where we can vaccinate someone against HIV. They may still become infected, but they can control the disease. So that's also a goal of HIV vaccination. And in that case, we're thinking that it may be the T cell response that may be most effective. So we think about the CD8 T cells as controlling infection. That cells are already infected, and then the CD8 cytotoxic T lymphocytes can kill those infected cells and thereby control the disease progression. So we're thinking both about T cells and antibody. Of course, we want both. We really would love to have both. And that's the ultimate goal. A few slides just talking about the work I do, even though I didn't really want to focus that on this talk because I want to keep it more broader, but I think it's relevant in terms of looking at the immune correlates from RV144. So we're looking at the T cell response to vaccines, and there are several ways of measuring the T cell response. One you may be familiar with is tetramer staining, but that's limited because you have to have the peptides or the epitopes selected ahead of time before you can actually look for those tetramer-specific responses. Interferon gamma ELISpot is a nice tool, and that is using ex vivo stimulation of PBMC, and then you're looking for cells that secrete the cytokine, and you're actually counting spots on a plate. That works well, but you're looking at just one cytokine, interferon gamma, and you're limited to looking at total PBMC, so you don't even know if those cells are CD4 cells or CD8 cells. Intracellular cytokine staining by flow cytometry is the technology that we use in the lab. It is an advance over ELISpot because it's multi-parametric, so it means that we can identify the type of T cell that's producing the cytokine, CD4 cells or CD8 cells or TFH cells, or we can even subdivide the CD4 and CD8 cells into memory classifications, so it's very flexible, and it allows us to look at multiple cytokines. We can look at Th1 cytokines, we can look at Th2 cytokines, we can look at Th17 cytokines, so we're able to broadly characterize the immune response in a lot of detail using flow cytometry. CFSA proliferation is another type of assay. I include that here only because sometimes that's used if you have a response that's so low level you cannot detect it with a direct ex vivo assay. With CFSC, you expand those antigen-specific cells over 6 days, and that can sometimes improve the sensitivity of your assay. So in terms of a few more details about the intracellular cytokine staining assay, we're mainly using peripheral blood mononuclear cells, but just so you're aware, we can also use other cell types. We're interested in mucosal immune responses, so in some of our studies, we're actually able to get gut biopsies, and we're able to isolate mononuclear cells from those gut biopsies and then look for antigen-specific cells in those responses. But most of the time, we're looking at peripheral blood mononuclear cells, and we stimulate those cells cells ex vivo for a very short period of time. Usually it's 6 hours. And usually we use peptide pools that cover the antigens of interest. And those are typically HIV proteins that are encoded in our vaccines. We have a negative control. In our case, because we're using peptide pools, the relative negative control is the diluent for those peptide pools, which is DMSO. And then results are reported as the percent of CD4 or CD8 cells producing the cytokine of interest. So for instance, if we have 0.5% of CD4 cells responding to a certain vaccine, that's actually a very good response compared to other vaccine candidates that were looked at. And it's also useful to come up with a positivity call for a response. And that's something I'll note that on the next slide or two because it relates later to a correlate that we're looking at in RV144. So even though it may seem like a rather sophisticated topic or maybe even just a rather simplistic topic, it's actually not that simplistic to come up with a positivity call that's very robust. And for us, what we're doing here in positivity is we're comparing the proportion of cells producing the cytokine of interest when we stimulate with an antigen, and we compare that to the unstimulated negative control. And typically, in the past, people have simplistically done threefold over the negative control. We prefer to use a statistical call, and one of the calls we use is based on the Fisher's exact test, comparing the actual number of cells in the positive stimulated sample versus the negative. And we have a new method referred to as MIMOSA that's a Bayesian method. I'm not going to show any details of that, but I will refer to it, I think, on the next slide, and 2 slides forward. When it comes to the polyfunctionality analysis, we're now using this method. This is the one data slide I'm going to show you for flow cytometry. I usually show a lot of these slides, but basically I just wanted to emphasize that we're looking at low-level responses. Each dot is a cell, and here we can see that we're looking at a response to an Ad5 hexon that happens to be one of the vaccine candidates we're using. And interferon gamma is shown on the y-axis, IL-2 is on the x-axis. Cells over here in this quadrant are co-producing interferon gamma and IL-2. These cells here are making interferon gamma without IL-2. And you can see there's a relatively small number of dots, but these are the dots, these are the responses that we want to measure. This slide happens to be comparing 2 of our positivity methods, showing that our older positivity method would not pick this up as positive, whereas our new MIMOSA method, which is more sensitive than Fisher's, is nicely picking this up as positive. By eyeballing the response, you can see it looks positive because it looks different than the negative control. So this MIMOSA method is really a nice method for increasing the sensitivity. And I bring up the MIMOSA because I wanted to follow up by discussing this other method referred to as COMPASS. COMPASS is just a way of looking at the polyfunctionality of the response. This term polyfunctionality has become very popular in recent years because there have been several studies that have shown that cells that are more polyfunctional are likely to be more protective. People have done that by looking at non-controller, I should say HIV controllers. These people who are infected with HIV and naturally control the infection have more of these polyfunctional CD8 T cells. So we feel it's important to look at polyfunctionality in the context of vaccination as well, because we will hypothesize that more effective vaccines will have more polyfunctional responses. So COMPASS is this new method that our colleagues Rafael Gottardo and Greg Finek have developed that uses the MIMOSA and extends the MIMOSA by applying that positivity separately to each polyfunctional profile. When I talk about a polyfunctional profile, you can imagine if you're looking at 5 different cytokines, you can have, I believe it's 32 different flavors of cells making or not making each of those combinations of cytokines. And this analysis performs that in an unbiased way, treats each of those profiles independently, and will give you a score And one of the nice aspects of this method is it gives you a single metric score that summarizes the polyfunctional response, either a functionality score or a polyfunctionality score, which you can think of as the proportion of all the antigen-specific responses that are detected among all of them, the ones that are detected compared to the total denominator. So functionality versus polyfunctionality score. Polyfunctionality score is similar, but it simply weighs the functions differently. And I will return to this in just a few slides in the RV144. So I spent a few minutes talking about this because this COMPASS analysis has revealed a cellular correlate in RV144. And we're now using this in our other vaccine studies. Okay, so a few minutes talking about the RV144 trial. This is such a seminal trial in the HIV vaccine field that I think it's important to focus on it and spend time on it, because we've learned some lessons from this trial that we're now carrying forward into future trials. So that's the reason I'm focusing a bit of attention here on this. The RV144 trial used a combination of a viral vector, this ALVAC, it's a canary pox vector, encoding GAG and protease, and that was administered in 4 doses. And then 2 of the doses were co-administered with a protein. It was referred to as AIDS Vax, GP120. So the protein vaccination is very similar to what we're familiar with, for instance, hepatitis B. It's very similar to hepatitis B vaccination, where you have the hepatitis B surface antigen with the alum adjuvant. That's very similar to what the AIDS Vax is. It's simply an HIV envelope protein with alum, but it's co-administered with the ALVAC. And there must be something about this co-administration that was critical to the success of this vaccine, because 2 previous efficacy trials using AIDS Vax alone showed no efficacy. So there's something about this combination of the canary pox with the AIDS Vax that was beneficial. We don't know exactly what that was, but empirically this is what we found. So the correlates were, the study, I should say, was first released in 2009, so it's a number of years now, but like I said, it's had such a critical effect on our field, I think it's important to highlight it here. Efficacy at the end of the study was only 31%, but I think it's important to point out that if you only look within the first year after vaccination, the efficacy was 60%. So we're feeling as though this vaccine was quite efficacious, it just didn't have longevity, it did not have durability. So that's another issue. We're designing new vaccine candidates that can improve the durability. One of the ways of doing that is to give booster vaccinations down the line so that you improve the durability. And then the correlates of risk, there were 2 primary correlates of risk. One was the antibody to this V1, V2 variable loop region of the envelope protein. And it was the IgG V1, V2. And that was a positive correlate, meaning that if you had high levels of antibodies, you had a decreased risk of infection. Paradoxically, we had a negative correlate, or what I refer to as a negative, this IgA response to envelope. And that was the opposite. Actually, if you had higher levels of that, you were more likely to be infected. And there's a little bit of a complicated story there where it really wasn't increasing your infection, but by comparison to the group that had lower Ig levels, it was different. So it's a little bit complicated to go into that, but there have been many studies following up on this, and I think the thought is that the IgA is interfering with the protective effect of the IgG. So there's some kind of interference there. And again, we don't totally understand what's going on with that yet, but they're continuing to go on with that. I did want to talk a little bit about the cellular immune variables. So I just told you that the 2 primary correlates are antibody correlates. But I'm going to also show you that we did find a cellular correlate. It was not detected in the primary analysis, but when we applied the COMPASS analysis in a secondary analysis, we did find a T-cell correlate. We, of course, used intracellular cytokine staining as the primary readout. We were looking for these 4 different functions here. And we also had another assay where we were looking at secreted cytokines, and that also showed to be a correlate in secondary analyses. This slide is just showing you actually what the original correlates analysis showed with the IgA and the IgG correlates. Those are the two correlates I just referred to before. Again, showing you that the relative risk is above 1 for the IgA, meaning that it actually was harmful, and the relative risk was below 1 for the V1, V2, which again was a protective correlate. And I'm not going to spend time talking about this, but as I mentioned, this study has really been seminal in the field, and there have been a large number of follow-up studies, many of them confirming the original correlates. So I think we all in the field feel very confident now that these correlates are true, and that other people have now followed up with in vitro studies trying to understand what could be going on with those. One of these studies here is this polyfunctional CD4 response, which I'll be talking about in a moment. In addition to the ICS response, I mentioned that we were also doing some cellular studies looking at secreted cytokines. And in fact, although not a correlate in the multivariate analysis, in a univariate analysis, we did find some of those cytokines to be correlates. This is just showing that IL-10, people that had higher levels of IL-10 were less likely to become infected. So, and then this is also looking at the ICS response. Again, people that had a total ICS cytokine response that was high were less likely to become infected. So this makes us feel comfortable that truly there also is a cellular correlate, and that it goes in the right direction, where if you have a higher cellular response, you have a lower infection rate. But what we were most intrigued by was our COMPASS analysis, because using the COMPASS analysis, we looked at all 6 functional markers in an unbiased way, and the result of the COMPASS analysis was that we were able to find a correlate that actually stood up even in a multivariate analysis. I mentioned briefly before that the COMPASS analysis reports 2 metrics, this functionality score and the polyfunctionality score, and that's what's graphed here. And what this graph is showing is that the people that became infected had lower functionality scores and lower polyfunctionality scores compared to those that were non-protective. So this is just a visual representation, but if you put it into a proper logistic regression analysis in combination with the primary correlates, this stood out as another correlate. So indeed, there's a CD4 correlate in the RV144 trial. It's interesting to think about why did we not detect this ahead of time when we were doing the full correlates analysis. And one of the reasons for that is we had some presupposed hypotheses, and we actually didn't even include IL-4 when we were originally doing the correlates analysis, whereas the COMPASS analysis was unbiased to include all the markers. So it also detected a subset that was very rare, and the magnitude is very small. But once again, the COMPASS analysis is not biased in any way. So I think that's one of the reasons we were able to detect this with the COMPASS, whereas in our predetermined bias types analyses, we missed it. And just to think about what could this potentially mean, I think it's reasonable to think that if you have an antibody correlate, you're likely also going to have a T-cell correlate that goes along with it, because you think you would presuppose that you would need CD4 help to help the B-cell response, and that is what we're finding. It is curious that it is a polyfunctionality response. I should say the T-cells are polyfunctional. As I mentioned before, we do think that polyfunctionality is better, and this actually happens to show that. And it's surprising, though, that this polyfunctionality subset doesn't fit the predetermined Th1, Th2 paradigm. It actually includes both interferon gamma and IL-4. So once again, it's something we may have missed if we had bias ahead of time in terms of what we were looking for. The bottom line of this is that we are now using this COMPASS analysis in all of our studies. And I'm sorry, I forgot to include the one slide for HVTN 505 where we used this analysis and did find another polyfunctional correlate in the HVTN 505 study. And that, again, hopefully should be coming out in print relatively soon. It's just been submitted for publication. Okay, then I'm just going to finish the rest of my talk talking about vaccine concepts. So I've talked a lot about RV144 because it's the only trial that we've had that's shown efficacy, and it's really helped us think about where to go next in terms of efficacy testing. But that's not the only vaccine concept we're testing. There are many other types of vaccine concepts that are in development and in clinical testing. So I thought I'd finish by going through some of those concepts because I think it's very useful as a medical audience to kind of know what's coming down the line. The very first bullet point here, we refer to it as the P5 program. It's this funny pox protein public-private partnership, but really all that is referring to is there's a lot of work being done following up on the RV144 trial. And I will finish up by talking about that because that's where we have our next efficacy trial that's just started. And that's as a result of the findings that were released in 2009. It's taken us this long to get to the point of the next testing. But we're very excited by that. So I'll finish up by talking about that. But there are other concepts that are being tested unrelated to the RV144 that I think it's useful to be aware of. One of the most interesting is this concept. We're returning to the concept of neutralizing antibodies. So the RV144 correlate was a non-neutralizing antibody. But there's been a lot of work in the last 5 to 8 years showing that indeed people who are infected with HIV do develop neutralizing antibodies that are broadly neutralizing to very many strains of HIV. Up until a decade ago, we didn't believe that was possible, or we believed that was so rare that it wouldn't be end of any use to try to develop such an antibody. But now it's been repeatedly shown that many people who are infected with HIV over the course of several years eventually develop these broadly neutralizing antibodies. And that's very exciting because that gives us hope that we can maybe develop vaccines that can induce those broadly neutralizing antibodies. So there are a number of concepts now in place where they are designing relatively complicated concepts, but where we're hoping to build on that and to actually try to develop vaccines that can end up with a neutralizing antibody response. So I'll tell you about one of those concepts. And I'll talk a few moments about that. T-cell vaccines. We haven't given up on T-cell vaccines. We call them T-cell vaccines. That's kind of an unusual name. What it means is vaccines that are mainly designed to induce a T-cell response rather than an antibody response. We call them T-cell vaccines. But many people have given up on that concept because they feel like it's only antibodies. Antibodies is the critical correlate. But obviously we feel, and most people do feel, that it's a combination of the two. So T-cell vaccines are moving along, and there's this really nice concept of mosaics that are in the clinic right now. And there's also a very nice concept of using CMV as the vector. And I'll just briefly mention those two. And they're very exciting concepts, and we're happy that both of those are just recently into the clinic now. And then, of course, we are interested in antibodies and T cells, so many vaccines are combining a T cell component and an antibody component. I won't show you any specific example of that, but a few of the phase 1 studies that are moving along are combining those. And we're actually collaborating with Janssen Pharmaceuticals to move into a phase 2 study where they're combining a T-cell vaccine and an antibody vaccine. So we're very happy to see those types of vaccine concepts moving into the clinic. And then lastly on this list, another exciting area of HIV prevention in recent years has been this idea that we can maybe prevent HIV infection using methods other than vaccines. And this, for the longest time, we were very discouraged by that because there were many studies that were looking at microbicides. For example, microbicides that are applied vaginally, but the initial studies were very, very discouraging. They were almost uniformly negative But then we started to get some positive results. We actually had a positive microbicide study, and then maybe even more significantly, this idea of PrEP, pre-exposure prophylaxis, where you take an antiviral drug, actually a combination of two drugs, orally on a regular basis, and studies have shown that that's extremely effective. So we now have renewed hope that there are other mechanisms of preventing HIV than just a vaccine. Of course, we still believe that in the long term, across globally, a vaccine is what's most critical need, because with these other mechanisms, it's really more expensive, it's harder to distribute. But in any case, we are trying to understand more about these other prevention modalities. And our own network is testing this antibody-mediated prevention. I told you that we've recently developed or discovered that there are broadly neutralizing antibodies. So the concept here is, can you infuse those broadly neutralizing antibodies into people who are not infected with HIV but at risk for becoming infected, and will that prevent infection? That's really a test of concept. Because keep in mind, what's strange about these broadly neutralizing antibodies is, I told you, they develop in people who are HIV infected, and they usually develop years into the infection, but they're pretty much of no use to those people. It doesn't help their disease. They develop them, but it really doesn't control their disease. So, we have to prove the concept that yes, even though it may not be useful in people that are already infected, maybe it'll be useful in preventing infection. And I should say that there is some thought about using these as therapy in people who are already infected. And perhaps if their body has not already developed resistance, there may be some use. But in terms of the people who are developing them, it doesn't seem to be of much benefit in those people. So there's still a lot to learn about these broadly neutralizing antibodies, but our network is testing them, and we're excited about this. This is a new concept that's just started testing within the last year. So moving on to just finish with a few specific examples for these. The induction of these broadly neutralizing antibodies. So I mentioned this is relatively new. Up until about 5 or 8 years ago, there were about 5 broadly neutralizing antibodies, and that was it. Now there are dozens. And it turns out that there are certain regions of the HIV envelope that these broadly neutralizing antibodies target. One of them here is the CD4 binding site, and that's one of the ones that we're testing within our network. But this is just, so they exist now. And there are certain characteristics of these broadly neutralizing antibodies that seem to distinguish them from non-broadly neutralizing antibodies. One of them is they have very high levels of somatic hypermutation. So you can think about, once we discovered that, that was a very discouraging thought from the perspective of vaccination, because it means that you need a lot of mutations happening in each person to get to that broadly neutralizing antibody. Which is a problem, because we want to really vaccinate someone, and then we would hope to have like a quick response to that vaccine. That's not going to happen. With the somatic hypermutation, it means that you have to have several rounds of mutation to get to that broadly neutralizing antibody. They generally have very long CDRH3 regions. Some are poly or self-reactive. We don't really know what that means yet, but interestingly, they start out perhaps by being cross-reactive with gut flora. And there's certain overrepresentation of certain germline variable heavy chain regions. And then what people have done is they've actually gone through and they've been able to analyze in detail how these antibodies develop, and then revert back to the germline sequence. And what's confusing and a little bit disheartening in terms of vaccine development is the germline sequences may not even bind directly to HIV. They actually may bind to some gut flora, and then as they somatically hypermutate, then they develop their reactivity against envelope. So again, very challenging when we start to see this, but people are working on it. So we've discovered that HIV and antibodies co-evolve over time. So there are many people that are actually doing these very sophisticated studies looking at the development of these broadly neutralizing antibodies with the goal of reproducing this development by a vaccination strategy. And one of those strategies, we're working with Bart Haynes at Duke, and he has designed a series of immunogens that could hopefully mimic the evolution of those broadly neutralizing antibodies over time. So this is a new concept that's about to start in the clinic. So we're very excited by this. Again, it's a complicated strategy, but we're hoping it will be a test of concept that you can start with sequential immunization with different envelope variants that will end up forcing the somatic hypermutation in the direction you want to go. So it's quite an elegant theory, and we'll see for the first time whether it works in humans, because this trial is just about ready to start. So again, a new type of concept, and we'll see. It's fascinating in my mind, and we should get some data soon. In terms of what's new in T-cell vaccines, you know, the issue with HIV is as we know, is it mutates rapidly, so you very often evade the immune response because HIV will just mutate away from it. So there's been a lot of thought about, well, is there a way of designing vaccines that are like broadly neutralizing antibodies that will develop T-cells that are broadly reactive to a lot of strains of HIV? So even if it mutates away, you'll have another epitope that you'll be able to respond to. So one of the ways this has been designed is statisticians have actually developed these mosaic vaccine structures that kind of capture as much of the variability as possible in these simple structures. And they're called mosaics because they're artificial immunogens that are put together on the computer. And again, it's a fascinating strategy to be able to do this computationally. And now we've actually pulled those into the clinic. And we have one of our studies that I actually have a little bit of data on. And it's for this HVTN-106 vaccine, where actually we're comparing this new vaccine, the mosaic vaccine, compared to a consensus vaccine, which is another thought in terms of how you can maybe take something that's a consensus of many different strains, and then comparing it to a single clade B naturally transmitted virus, and seeing if is the mosaic as good or better than those. This particular vaccine uses a DNA prime with an MVA that's modified Vaccinia ankara. It's a pox virus boost, but we're really focusing in this study on the differences in the DNA prime. And interestingly, it's kind of complicated to see, but what we're looking at here is we're looking at T1 is the natural envelope virus. Treatment group T2 is that consensus, and treatment group T3 is the mosaic. And what you can see compared to the natural, this is the CD4 response rate. 50% of individuals responded, whereas 72 responded with the mosaic. So we are at least getting an increased response rate, and it actually looks similar to the consensus. So encouraging results. Again, this is really the first test in humans of this vaccine concept. So very encouraging that this is moving forward. So it's nice to see that there are new concepts. And again, I think in the end, it's probably going to be a combination of these concepts that are going to work. It's probably not going to be any one on its own. And then this is one data slide from Louis Picker in Oregon, who's doing these really fascinating studies in non-human primates where he's using CMV as a vector. So many of our vaccines are using viral vectors, and they're encoding DNA genes into them for HIV, and he's using CMV. And it turns out that CMV has some unique characteristics, and his vaccines are showing something that we've just never seen before, where he is seeing essentially complete protection. And in some cases, it may be sterilizing immunity, but in most cases, it's not. Whereas the animals are becoming infected with HIV, but they're controlling the disease. This is viral load shown here in red, and this is a group of monkeys that have been vaccinated with his CMV construct, and every now and then they have a tiny blip indicating that they're infected, but they're not having disease. So this is a concept that's really fascinating, and for the first time that's going to be coming into the clinic, probably in the human testing, in the next year or so. There are obviously issues about CMV. CMV can cause human disease, so there are a number of factors in terms of bringing this into the clinic, but they are working on this, and I just wanted to highlight this as another new concept that is very fascinating. It's just showing different results than we've seen before, so some encouraging news. And then I'm going to finish up the AMP study. We call it AMP, antibody-mediated prevention, and it's using this VRC01 broadly neutralizing antibody being infused into people passively to see if it prevents infection. An extremely complicated study, because when I say infused, it literally means to be infused. You have to put an IV, and the participant has to sit there for an hour getting an infusion once every 2 months. So this is an extensive study, and we're very proud of our clinical sites for being able to do this. This is being done in America, and it's also being done in several sites in sub-Saharan Africa. It's just started this past year, so it'll be another year or two before we get results. But again, it's a test of concept, and it'd be great to see, can this actually work? And we're excited by that. And then lastly, I'm almost fine. I just have like 2 slides on RV144. So the follow-up on RV144 is that RV144 was this clinical trial that was conducted in Thailand, and it was using these combination of vectors that were for clade A, clade E, the type of clade virus that's circulating in Thailand. What we decided, and what the field decided, is that it would be most useful to see, can we adapt this for the type of virus that's circulating in sub-Saharan Africa, because that's where we have the biggest epidemic. So we decided to embark on a number of different studies. So the first study that was conducted was, let's just see, can we administer the same vaccines in a South African population and get similar immunogenicity? And that was done in HVTN-097, and indeed we did get similar immunogenicity, in fact a little bit better. So we feel as though the population is not going to affect the ability to respond to the vaccine. Then what took a really long time is that we had to work with sponsors, the companies that develop the vaccines, to create new vaccines with clade C inserts for the viruses that circulate in South Africa. That took years. It was discouraging how long it took, but just the year before last, the products were ready, and HVTN was a phase 1 study testing the immunogenicity in South Africa. We conducted those studies within the HVTN, and the results look great. Immunogenicity was comparable to what we saw with RV144. So that has given us the green light to go forward with a new efficacy trial. We're really excited by this. The first participant was enrolled, I think it was just 2 weeks ago. We actually haven't released it publicly yet. We're going to give a press release on December 1st, World AIDS Day, about this new clinical trial, new efficacy trial, using the same type of regimen that was used in RV144, but in South Africa. Luckily, and again I say luckily, it's a problem, but because the infection rate is so huge in sub-Saharan Africa, that we can design a relatively small efficacy trial to get a very clear answer. It still will take several years before we get the signal, but this is just started, and we're very excited about it, and that is happening. And actually, Tony Fauci released this quote about how this is the first time in 7 years that we finally have another large-scale efficacy trial for HIV. So I think that's a nice thought to end on, that we are moving forward. Just acknowledging, I work at the Fred Hutch. Julie McElrath is my mentor. Larry Corey is the director of our HVTN. My colleagues at the Hutch, Georgia Tamaris at Duke, who's a very close colleague. And then our funding comes primarily through the Division of AIDS, but we also get funded through the Gates Foundation, and I get some funding through the CFAR. And we're always very grateful to our statistical colleagues within the HVTN and all of the trial teams that work to get these trials. It's a huge effort, and of course all of our vaccine trial participants. Okay, so I'll end there. Thank you. There's a question over there. I think we have a microphone. Oh, there, thank you.
Thank you for an interesting talk. I wonder if you could comment about the HLA haplotypes of the vaccine trial participants in the RV144 that showed some efficacy, and does that either confound one's conclusions or help explain the conclusions for why some efficacy was observed?
So there was genetic testing done, and the HLA types were analyzed. And if I remember correctly, and I may be incorrect about this, we did do an analysis trying to see if there was any correlation with HLA type and the response to and the correlate, I should say. And for the most part, I believe it was not correlating. There was some small responses, some significant but borderline significant responses, I think, with class 2 and also with the NK kind of cure responses. And I don't remember the details of those, but in any correlates analysis, they do also look for genetic testing. And as you can imagine, that's very important to see if there is any correlate. But I think for RV144, I don't think there were many significant findings, but it was looked at. Yes.
So then when you apply this interesting compass analysis to the trials where there was no efficacy, do you see any significant responses, for example, in the HVTN505 study?
So what's interesting, and I was discussing this with Amalea a moment ago, so with HVTN505, it was a relatively large study, and that happened to be an efficacy study, so we can actually look for, people who became infected and did not become infected. And we did see an interesting signal with the compass where polyfunctional CD8 responses to envelope, if you had higher levels of those, you were less likely to be infected. But the curious factor there is because there was no overall infectivity signal, it must mean that if you have lower responses, you're more likely to become infected. So there's that balance there that if you have no efficacy signal, you may have an increased risk versus a decreased risk. And I don't understand the statistical details of it, and I should have reviewed that manuscript before I came here today. But we did find that that's the one other case where we've seen an efficacy signal with compass. Those are the only two so far that we've looked at.
So you have this great slide that showed CD4s and their critical role being central in developing productive immunity, both for antibody responses and CD8 responses. So at the beginning of your talk, you talked about, various stages of disease that the vaccine trial participants currently have when they get vaccinated. So could you help a lot of people in the audience here understand how you use a vaccine in a disease where you destroy CD4 T cells that's really central to getting an efficacious vaccine response?
Oh, yeah, so all of our studies are prophylactic. They're all in HIV uninfected individuals. So I didn't emphasize that. The HVTN only does that. But there is a large interest in using similar vaccines as therapeutic vaccines. Our network does not do that testing, but there is interest in that. And then, as you said, there has to be a lot of thought put into what would be the best candidate. And in that case, most likely it would be candidates who are not so severely immunocompromised so that they're able to develop an immune response. That's a complicating factor in therapeutic vaccines.
Steve, thanks for the great talk. I'm curious about what your thoughts about what has led to the renaissance in neutralizing antibodies. Is it just better techniques, or were people just not thinking about things the right way?
I think a big part of it is the techniques. I think there are even people within our own group, David Montefiore at Duke has developed these panels of antibodies that make it very easy to screen for responses across different strains of HIV. I think that's a big part of it. And I think, as you referred to, I think people were just not thinking they were going to be there. So why should we look for them? That's my guess. Again, this is not an area where I'm intimately involved because I don't work in that area, so we'd probably have to ask an expert in that area. But I think you highlight the two primary reasons. With Chihiro, do you want to go?
Hi, thanks, Steve. That was a great talk. So my question is, are there any characteristics of the T cells, these polyfunctional T cells, that in terms of activation markers or, memory subsets that mark them as polyfunctional, or do you really have to do the intracellular staining and that's all you can do to identify them?
So far, we haven't detected any, but we actually have a study that we're just doing in the lab right now where we're sorting polyfunctional and less functional T cells and looking at RNA sequencing to help us see if we can determine what's different about them. But yeah, because we have not identified any other way, any other unique characteristic of them. So you highlight something that we're very eagerly looking at in the lab right now. So, I think we had a question here first. Okay.
Thanks for the talk. I have to say I recognize some of the names of strains used in the vaccine reagents from graduate school 10 years ago. So there just seems to be a very different approach towards choosing vaccine candidates than, say, in flu, where we're surveilling populations and choosing antigens to match. Can you explain and maybe put into context why we don't seem to have that same approach in HIV vaccine design?
I can't really explain with a lot of expertise, but I think one major factor is that it takes a long time to get vaccine candidates into the clinic. They have to start development, and there's so many phases. I think it's a little bit different than with flu that, it may take 10 years. So I think, unfortunately, some of the strains, some of these vaccine candidates were originally thought of 10 years ago. So I think that's an issue. I think that it is one of the factors. But the more complicated issue in terms of how do you best determine what strains to start with, that I couldn't answer, and that I'd have to refer to the experts. And maybe that's why the mosaic vaccines are a great way of sort of getting beyond that bottleneck. I think there was one question over there. Yeah.
So I'm curious if, so the sort of need for polyfunctionality seems to go along with some of the work from Galit Alter, who's looked at polyfunctional antibody responses.
Yes, right.
I think in the same, comparing these same trials that you've talked about, the ones where there's efficacy and not, and she sees a correlate between polyfunctional antibodies and efficacy. And so I'm wondering if there's something just on a very basic level about antibodies that do more things and T cells that do more things go together, kind of, and whether you can put that data together with your data to sort of broaden the analysis.
Yeah, no, we work closely with Galit Alter, but we have not pulled those data together yet, and that's one of the issues. Is we have a shortage of statistical help. Actually, we've been developing a lot of data within our network, and we have a lot of different parts of our network, a lot of different labs, and honestly, that's been a challenge for us to pull together all the different parts. But you highlight one area that we haven't done. We haven't pulled together a lot of the work that Galit has done in other labs with the T cell work. No, it hasn't been done yet.
Thank you. I was wondering about the neutralizing antibody. You were saying that as different vaccines with that response are implemented or targeting that particular response, that response might take time to develop. And so I wondered if it was contrary to the case for RV144 where the vaccine efficacy lagged, that maybe the vaccine efficacy would actually increase over time.
Yes.
Is that something that people are anticipating, and will that affect the statistical design of the the trial analysis?
I'm sure it will. Of course, these trials are clearly in phase 1 at this point, so they're not reaching that point yet. But that is going to have to be thought of, and it's a major issue as well. Is that if we're going to be putting these vaccines into people that are actively at risk for HIV, we don't want to wait that long. So most likely those trial designs are, in my mind, you're going to have to include some other kinds of prevention during that ramp-up period for those developed. So I would think they should be combined with PrEP or some other, or antibody-mediated infusions while those antibodies are being developed. So it's a complicated, it's not easy, and you're right, statistically we're going to think about the design of those trials. Let's hope we reach the efficacy testing with those vaccine candidates at some point. We can just be hopeful. Okay. Thank you.